Hi, this is Dr.P.Thirunavukkarasu from Dr. MGR Educational Research Institute, Department of Biotechnology, Madurai, Chennai. In this video, I want to talk about eukaryotic transcription initiation process. So in this video, following content, I am going to explain. First one is definition and introductions. What is the difference between prokaryotes and eukaryotes? What are the steps involved in the transcription process? Initiation, elongation, termination, regulation of the transcription and mechanism of the transcription. What are the requirements of the transcription, such as the start site recognition proteins, recruiting proteins, regulatory proteins. So regulatory proteins is divided into two. One is activator and repressor. First, what is transcription? Transcription has been defined in various ways. The synthesis of RNA from a single strand of DNA molecule in the presence of enzyme RNA polymerase is called transcription. In other way, the process of formation of messenger RNA molecule using a DNA molecule as a template is referred to as transcription. Next one is introduction. Eukaryotic transcription is more complex compared to prokaryotic transcription. In what way the eukaryotic transcription is complex? During the process of transcription, there is two components of RNA such as intron and exon. In prokaryotes, there is no intron and exon. In eukaryote, one promoter is controlled with one gene is called monocystronic expression. In prokaryote, multiple genes controlled with one promoter and operator is called polycystronic expression. So the eukaryotic transcription is more difficult to start initiation. Prokaryote initiation is very simple compared to eukaryotes. In this process, sigma vector find to promoter region and start initiation. The sigma factor joined with some other factors such as the 2 alpha, beta and beta dash. This unit is called as holoenzyme and core enzyme. In prokaryote, there is only one RNA polymerase responsible to transcription process, but in the eukaryote, there are three classes of RNA polymerase involved in the transcription process, such as RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Eukaryote initiation process involved in more transcription factors, more proteins and more enzyme. For our understanding, we have divided into three parts of transcription, such as initiation, elongation and termination. The eukaryote initiation, there is a two part. One is mechanism of the transcription and regulation of the transcription. So what are the requirements of the transcription? So there are start site recognition proteins, recruiting proteins and regulatory proteins. So what is start site? Start site is also known as promoter regions. The promoter regions, there is unique sequence. The RNA polymerase, which is major enzyme to identify the promoter region. This is the double-stranded DNA. This double-stranded DNA have promoter regions from -35 to -1. This promoter region itself Tata box regions. So this is known as start site. In this process, mechanism of the process, transcription factor 2D and transcription factor 2A and B RNA polymerase transcription factor F, transcription factor 2, E and H. These are all the protein involved in the transcription process. So the first one is transcription factor 2D is first bind in the double stranded DNA. The transcription factor 2D carry with two more proteins, that is transcription Tata box associated factor, Tata box binding proteins. So it is known as recognition proteins. After binding of transcription factor 2D to Tata box, it modifies the DNA structure. Once binding the transcription factor 2D, another proteins is going to bind in the promoter regions. So that is known as transcription factor 2A and B. It is known as recruiting proteins. It is helps to RNA polymerase properly recruit the two promoter regions. In the third step, RNA polymerase is going to bind in the promoter region. But RNA polymerase cannot bind with alone. It needs escort proteins. That is known as transcription factor F. This remain attached to RNA polymerase. This F protein have major role in the transcription process. So it is bring the RNA and guide the RNA to proper binding of the start site. This stage is called free initiation complex. In this particular stage, RNA polymerase plays in the start site, but it is waiting for proper signal. This is the point of regulation of the transcription. If positive signal is there by the activator regulatory proteins, RNA polymerase will escape from promoter and initiation will start. If negative signal is there by the repressor regulatory proteins, RNA polymerase cannot escape from promoter. So RNA polymerase remain attached to the promoter regions. If there is positive signal, another transcription factor going to bind to the promoter, that is known as transcription factor E and H. Still the promoter is not open, DNA is not melting, RNA polymerase is not escape from promoter regions. We want to open a promoter region and we want DNA melting and RNA polymerase escape from promoter regions. So this function done with transcription factor E and H. The transcription factor E and H have versatile activities. It have two functions. One is helicase and another one is kinase. Helicase always unwind the DNA and open the promoter region. The protein have two sites. One is C terminal and another one is N terminal. So always the C terminal, the phosphate group is attached. This phosphate group activated in the H proteins and phosphorylation of the process. So the promoter will escape and the polymerase also move towards the DNA strand. Here ATP's hydrolysis to ADP. So one phosphate is utilized. So the initiation is started now. So this is the overview of eukaryotic transcription initiation. Thank you.